Hi, thank you for having me. I'm the new director of a new Office of Nutrition Research, and I thought when ASN asked me to come in and give a talk, I would be talking about our office. And they asked me later to talk about these changes in rigor and reproducibility in the grant application. So I went over and talked to some of the SROs and said to them, do you know anything about these changes in the grant applications that are happening? And most of them told me to go away. So, but that was about 4 weeks ago. By now, most of them have had considerable training in the changes that are happening in the NIH grants. And if you didn't put a grant in the last cycle and were planning one for this next cycle, this is something that you'll really have to pay some attention to. These slides are available online. They're from ASN. There's a contact person here, reproducibility at nih.com. I tried this email address. I wrote to them and said, does this really work? And they wrote right back to me. And so the person who's in charge of this effort can help you answer questions. And there's quite a bit of really good training. And a lot of the training focuses, is somewhat defensive. It focuses on, what were the things that happened that caused NIH to make these changes in the grant applications. They just didn't wake up one day and decide to make some changes in the grants. They came from a confluence of issues, some of which we've heard here already, but actually a number of other issues that different institute directors became concerned about, and as these other problems arose, came together to make these rather large changes in the NIH grants. So these changes, these problems included worries from the Office of Women's Health Research that very few studies were, preclinical studies were being done on female mice, and female rodents. They were being done mostly on male animals. There's been a worry by John Loesch at GM that many of the cell lines that people claim that they say that they're using in papers are not, the cells that they thought that they were, and this has led to problems in reproducibility in research. And there's been, for a number of important disease targets, preclinical research for ALS and cancer where studies have not been reproducible. There's many people who've developed animal models or transgenic animals where an obesity claim was made, and when the drug companies obtained the animals or licensed the animals, they haven't been able to reproduce the results. So, something has to be done about this. Frequently people say, why doesn't NIH do something about this? And so, a number of changes are being made. Now, to improve reproducibility and rigor and transparency, a number of things occurred. Just didn't wake up one morning and did this. They conducted a number of workshops to address these problems of cell line problems and using, considering sex in outcomes of research. There was a 2014 workshop with journal editors that came to a consensus on reporting principles for preclinical research, which is similar to the kind of principles that we use for clinical research. There were 30 journals involved. They developed a checklist to ensure reporting of important experimental parameters. They sort of all have now in their instructions the policies for statistical analysis and how they'll ensure statistical accuracy. And one of the findings conclusions from this workshop that came out was sort of a reminder that reproducibility, rigor, and transparency, along with independent verification and validation of key cornerstones of the scientific method. So, that's one thing. And then they've developed a number of pilot programs to study. They just didn't make these changes in the grant applications. They did studies to examine the ramifications and feasibility of changing the grant applications. Could they do this without making the page limit longer and things like that? And they developed the training programs that you can receive and that the SROs and other people internally receive. And so these are some of the program announcements, some of the pilot programs they did here to look at the changes in the biosketch, checklist reporting guidelines, training, evaluation of scientific premise in grant applications. We've been seeing here a number of, the Biochemistry Society is looking at these approaches to reduce perverse incentives in research and efforts, FOAs, to support replication studies. And then there's been this, I don't know if you're aware of this, the PubMed Commons thing, where when your abstract is in PubMed, people can write something about it that they didn't like the statistics in it. And it's PubMed is more social media, starting to have more of a social media feel, and so hopefully my papers don't become subject to that. So changes to the grant proposals will be in 4 key areas. And so I don't really have time to go through all the historical things that led to these changes, and I'm going to just try and, as if my medical students when I was teaching in medical school didn't want to hear the History Channel, they just wanted to know what was going to be on the exam. So these are the 4 areas that you'll have to address. Scientific premise, rigorous experimental design, consider of relevant biological variables such as sex, and authentication of key biological and chemical resources. Let's just go through these. The first is a scientific premise. So the applicants will be asked and should consider scientific premise of their proposed research. You will discuss the strengths and weaknesses of prior research crucial to support the application, including whether the authors of those papers paid attention to the rigor, whether they were rigorous studies, whether their methods were valid, the analysis interpretation of the results were valid, did they use blinding and things like that in their approaches, did they have relevant biological variables such as sex, and did they authenticate key resources such as antibodies, or whether the Cree mouse they were using was expressing in the right tissue and things like that, or even expressing at all. Addressing the scientific premise is not meant to impede innovation. We want you to help identify the risks and develop rigorous research strategies to address the scientific premise that will bolster the success of your proposed work. Doing this should not interfere with exploratory research that frequently requires less discussion of the preliminary data or prior research. However, our rationale needs to be provided for your proposed idea. That should be clearly explained. And an important thing is to support the progression of science at an appropriate pace. We want to build, we don't want to build a large building on a poor foundation of research. So that's a key thing here. And so the instructions, your instructions about this, are sort of shown here in red, so that they're changing this will be in the research strategy. In the significance section, you'll describe the scientific premise for the proposed project, including consideration of the strengths and weaknesses of the published research or preliminary data crucial to support your application. Now, people have been asking me, well, how are the reviewers being instructed? And so the reviewer's instruction is here. So if you're on a study section, you'll get this instruction, and you'll be instructed by your SRO to address whether there's a strong scientific premise for the project, or is it built on a poor foundation. Now the next thing is, that's probably the biggest thing, is rigorous experimental design. And for preclinical researchers, this may be a big change from how they've been operating before. Frequently studies are done not based on power, but based on how many mice you had in the litter this time. So here we want a strict application of the scientific method to ensure robust and unbiased experimental design, methodology, analysis, interpretation, and results reporting. Full transparency in reporting permits others to reproduce and extend the research. As I said, we had this workshop with the journals, and the journal guidelines are, for most of the journals, should be aligned with the application instructions to reinforce transparency. They should be providing you more room to give the information about your methods and your biological resources, in the papers. Robust and unbiased results are reproducible under well-controlled and reported experimental conditions. So examples of rigorous experimental design to report include the use of standards, for example, in Western blots or something like that, sample size estimation, power calculations, randomization, blinding approaches. If you're taking tissues out of animals, perhaps people down in the next lab from you can blind the samples for you, and maybe you can have a relationship with the person who's in the next lab, and when you're taking tissues, when they're taking tissues, you can do that for them, and vice versa. Controlling for inter-operator variability. Statistical methods planned, and so rather than finding the statistic that shows that your results are statistically significant, devising a plan ahead of time for what statistical test is appropriate and what we will use. And and the preclinical researchers should reflect on whether, like the clinical researchers, they should put a biostatistician on their grant to help them with this thing. I'm sure David would agree with that. And then inclusion and exclusion criteria, subject retention and attrition, and how missing data will be handled, and other things that are appropriate to the particular kind of research you're doing. So here, your instructions for this are much simpler than they appear. Describe the experimental design and methods proposed and how they will achieve robust and unbiased results. In my opinion, this is actually going to be the thing that you'll have to think most about as you're planning your research. And for the reviewers, they're being instructed to ask whether the investigators presented strategies to ensure a robust and unbiased approach as appropriate for the work proposed. In many cases, you might be able to just describe this for one kind or experiment you're doing, so that they see your thought process, that might apply then to the rest of the aims that you're going to do. The third change in the grants is consideration of relevant biological variables such as sex. So this slide shows that much of the preclinical research on either has been on male animals and cells derived from them. And so Dr. Collins and Janine Clayton became very interested in this. And so part of the rollout of these changes came from them. The Office of Research on Women's Health has these 4 C's, so we have these 4 changes in the grants, and so 4 keeps coming up. And so the 4 C's of studying sex to strengthen science are to consider taking sex into account when you design your study, collect and tabulate sex-based data, characterize and analyze the sex-based data, and then communicate and report or publish sex-based data in your studies. And the last part of these changes are updating relevant biological variables. And so down here at the bottom, it's explain how relevant biological variables such as sex are factored into the research designs and analysis for your studies in vertebrate animals and humans. For example, there should be strong justification for the scientific literature, preliminary data, or other relevant considerations must be provided for applications proposing to study only one sex, for example, if you're studying prostate cancer or something. And then they have this note to further consideration of NIH expectations about sex as a biological variable. And then these are the reviewer instructions. Have the investigators presented strategies to ensure a robust and unbiased approach as appropriate for the work proposed. And have the investigators presented adequate plans to address relevant biological variables such as sex for the studies in vertebrate animals or human subjects. And the last change is authentication of key biological or chemical resources. It's recognized that these may differ from laboratory to laboratory over time. They have qualities or qualifications that could influence the results. For instance, the Cree knockout mouse you're using, is it still expressing Cree anymore? Those kind of things are clearly integral to the proposed research. So examples of things that you would need to authenticate are cell lines. Are they really 3T3L1 cells, specialty chemicals, antibodies, and other biologics. It's recognized that there's a consensus authentication guidelines are needed for many crucial resources. Many are universities. Your Office of Research Affairs may already have started working on things where you can send your cell lines someplace to get sequences done to prove that they're the cell lines that you say they are, whether they're male or female and things like that. They may be working on those problems for you. Researchers should transparently report how they will authenticate the key resources in the grant, and best practices will emerge from continued discussion and deliberation about this. And so this is a very long section. This is going to be in a separate attachment. It's not going to affect your 12 pages. And so you'll briefly describe methods to ensure the identity and validation of key biological or chemical resources in the proposed study. And the reviewers' instructions are that for projects involving key biological or chemical resources, reviewers will comment on the brief plans proposed for validating and ensuring the validity of those resources. And so the changes are sort of outlined here, the 4 changes: scientific premise, that you will be addressing that in the significance section. Scientific rigor will be a section that you'll do in the approach. And then consideration of relevant biological variables such as sex, that will also be in the research strategy. So if you're currently, remember that when you put in the animal research plan, that usually had 5 points. One of the points about how many animals there are and the sex of the animals, that will be moving now into your research strategies. And then the authentication of key biological resources will be a separate attachment, just like the sharing plan and things like that. So the page limits will not change. NIH has done studies that indicate that changing the page limits is not necessary. They did do pilot studies to do that. In terms of the implementation, if you submitted a grant last cycle, the changes that you had were already in there. The SROs are mostly trained now, and the peer reviewer guidelines are available on review procedures, and so the grants that are reviewed in June and July will have this updated language and have new review forms for the reviewers. So NIH expects that clearly designed experimental design and methods and applications and publications. In your application, you should address how the proposed research will achieve robust and unbiased results, and we hope that there'll be improved dialogue among members of the scientific community about a scientific resource rigor. This will be an important part of the quality control in our work that can be used to justify the trust that we want the public to have in us. That's it. Thank you.
Thank you very much. this is a serious question, not meant to be facetious, but your last slide said that you were going to use the same page limits. Did you have pilot researchers, highly skilled grantsmen or women, try and write grants within the guidelines before they were published to see whether the page limit criteria will actually be easily done? And the other was about the journal alignment. You said we want to align journal to the application. Does that mean that journals should actually increase their page limits?
Yes.
For all of the, now how are you going to impose?
We're not going to impose it. They had a meeting with them and they agreed to themselves to make these changes. You know, journals, it's actually the journals that have the highest impact factor are the ones where reproducibility has been the biggest problem. And so, and they know that they need to address this frequently, and so I think that they will be done in supplements and things like that. But all the journals, including this society's journals, are you know have agreed to that.
Could you answer the first question?
Yeah.
Thank you.
Whether it's been piloted.
It has been piloted, and so they had a funding opportunity announcement. They brought people in to study this question, and it was felt that this could be addressed in the page limits.
Andrew Brown, University of Alabama at Birmingham. I had the honor of serving on a series of roundtables as a representative for the American Society for Nutrition for FASIB on enhancing research reproducibility. And I just wanted to let everybody know that there was a document that came out in January because the NIH really did want it to be very community driven. And so FASIB has created a series of guidelines. It's called Enhancing Research Reproducibility that came out in early January that really should help people be able to follow along with some of the great comments that you've given today. So I don't know if there's a way we can distribute that again through the society, but I think it would really reinforce what you've presented to us today. So thank you.
Cheers. Thank you. Okay.